Hi, I am Mohammed Mortada, and today I'll be presenting my research on characterizing the immune response of broilers to Campylobacter jejuni. Campylobacter is the #1 cause of gastroenteritis worldwide, which is the infection of the intestine. How can humans get infected with this bacteria? Handling or consumption of raw or undercooked poultry meat represents the highest risk of infection in humans. What is interesting about Campylobacter is the different disease outcomes when comparing humans to chickens. In humans, the bacteria is highly infectious and could be detrimental for immunocompromised individuals. However, in chickens, the bacteria colonizes the chicken gut at very high numbers in the ceca and is asymptomatic. So the objectives of our study were to characterize the immune response of broilers to Campylobacter jejuni and to understand the responses that are responsible for the highly colonizing nature of Campylobacter in chickens. Day-old broilers were commingled until 2 weeks of age. At 2 weeks of age, chicks were weigh matched and were either challenged with log 8 CFUs per ml of Campylobacter jejuni or mock challenged with saline. Chicks were then raised until day 35 and organs were collected at 6 different time points. Campylobacter jejuni challenge did not affect body weight gain, feed intake, or FCR. The bacteria was able to colonize and persist in the ceca of chickens, where it reached around 5 log 10 CFUs per gram on day 35. Also, Campylobacter was able to disseminate to the spleen and liver. However, it was cleared on day 35 from these two internal organs. To validate our plating results, we measured Campylobacter jejuni MAP-A gene levels. And as you can see, pre-challenge, both treatments had similar MAP-A gene levels. However, post-challenge, the challenged group had a significantly higher MAP-A gene levels. On days 28 and 35, Campylobacter challenge treatment had a significantly higher serum IgY antibodies. This could support the bacterial clearance from internal organs seen in the previous slide. Also, in support of literature, we found that on day 0, there were Campylobacter-specific maternally derived antibodies. For bile IgA antibody levels, Campylobacter jejuni challenge treatment had a significantly higher IgA levels on day 21. Campylobacter jejuni challenge initially downregulated pro-inflammatory cytokines in the cecal tonsils, and through time, Campylobacter challenge upregulated both pro- and anti-inflammatory cytokines in the spleen and cecal tonsils. It is worth noting that the upregulation of IL-10 in the cecal tonsils supports the bacterial persistence of Campylobacter in the ceca, since IL-10 is known for its immunoregulatory functions. Attached splenocytes from both treatments were either stimulated with Salmonella enteritidis LPS or lysed Campylobacter jejuni and then incubated for 48 hours, where the nitric oxide production levels were measured using grease assay. On day 17, splenocytes derived from Campylobacter challenge treatment had a significantly higher nitric oxide production levels. Con-A is a plant mitogen that is known to stimulate T-cell proliferation. Therefore, mononuclear cells from the cecal tonsils were stimulated with Con-A or Salmonella enteritidis OMPs or lysed Campylobacter jejuni, and then were incubated for 72 hours, and T-cell proliferation was measured using MTT assay. On day 17, mononuclear cells derived from the cecal tonsils of challenged birds and stimulated with either Con-A or lysed Campylobacter jejuni were significantly suppressed. And on day 21, we showed that this suppression is antigen-specific, where it was only seen in the diluted Campylobacter jejuni. Our study opens the floor for many unanswered research questions. Is Campylobacter a commensal or not? What is the role of regulatory T cells? And how can we utilize our basic immunology in the development of successful interventions?